when we are talking about biology, we are working with quite complicated like organized structures, and only just simple example of growth hormone action, we can understand like a bit complexity of like data and how we can extract some additional value from in comparison of pathway analysis in our researches. So just like example, growth hormone is some substance, some peptide which like synthesized by your pituitary glands in your brain, and then it's distributed through blood flow and different tissues, and mainly like liver and muscles. They have like huge number of receptors for it, and growth hormone binds to them and stimulates protein synthesis through JK2 pathway and phosphoinositide 3K pathway. It's like increased synthesis of proteins, and one of them like main targets is IGF-1, so it's it directly affects transcription. And when we are talking about like biological regulations, we always have to like remember that we have working with DNA, RNA, protein central dogma when like, and then we have like loop when protein affects DNA transcription. And now we are today we'll be talking mainly about RNA and transcription, as it's kind of a relatively cheap and good marker of cell state and what is going on in the organism of cell. If we see that like some genes upregulated or downregulated, we can sometimes predict what is going on there and how it can be working. And when we are looking on this pathway, its complexity huge, and actually not all connections showed here, because pathways like it's never then some target hits another one, and they're involved more or less in other pathways. And it's overall this pathways that has some mutual space between genes and proteins. And when we are talking about growth hormone, it has like a huge like acceleration in protein synthesis and protein and growth of cells and like overall organism. And but just going back to the RNA-seq. What is RNA-seq? Basically, when we are talking about RNA-seq, we are thinking about some biological tissue or sample which was like homogenized in some way, and RNA was extracted from it. From RNA, we obtain like some cDNA, and then we synthesize this DNA, sequence this cDNA in sequence machine, like Illumina, and we obtained like some short reads for every like gene. And there could be a bunch of different reads, and we can map them on reference genes, like when we are using a reference genome. And for different genes in different samples, there will be like different number of reads mapped, and a proportion of these reads, it's kind of interesting for us, because the more reads we have for some gene, and when we normalize it for like for overall number of reads and for length of the gene, we can like talk about its counts. But even more interesting it becomes when we comparing two samples with like different conditions. For example, sample 1 is like control sample, and sample 2 is sample with some pathology. And when we are combining, when we have many people with control and with pathology data for genes, we can actually calculate if they have different expression in these genes. Why is this needed? Because we are people, and we have like different story, and our parents have different story. For example, if your mother was like fasting or during like pregnancy, it could be not so good for you, and actually this undertaking of nutrients, it could affect you in the future, and you will be more prone to diabetes, and maybe even your children will be more prone to diabetes. And for example, like some offspring from parents, from fathers who like, conceive later in life. They have longer telomeres, and it also like affects the bench stuff. And so we have like all of this like noise from different genome, like we have different mutations, and we have different epigenome, and we have like some exposome when we like through our life, when we have early childhood, some traumas, etc. It also affects overall our life, and it has long-lasting effects. And that's why we need a huge number of patients in every group, in healthy group and in control group, and in a disease group, and then we can extract mostly affected genes. It's a bit easier when we are working with cell lines. Most of drugs, they are going through this stage of cell lines testing when you add in your drug to cell line in a petri dish, and you see if like some changes occurred, some like changes which want to occur. So basically, when we are comparing treated and untreated samples, we can also calculate these differential expression signatures, and it will require a lower amount of samples, as they are like more even in terms of genetics and exposure and like their previous history. And for different drugs, you can like accumulate this number of signatures for different cell lines, for like different cancer lines. And when you have disease and you have this signature, you can try to find some drugs which will be able to like treat it, to treat this disease. And it's like it's a field which is now like quite growing up quite fast, and like we are doing some research in this area. And also it's quite important to find targets for gene therapy, because we have, and when we are talking about gene therapy, if we affect some gene which are like at the top of the pathway, it has a lot of connections, we will be able to like provide significant upregulation or downregulation or affecting the pathway, and it could be like we can be interested in this. But sometimes maybe we want to try to find some genes which are closer to our like point of interest, in order to like disrupt to not disrupt expression levels of other genes in the pathway. And it's quite important, because now gene therapy is like on its in anti-aging, for example, it's only developing, and we have like few like targets which are well known for like a long time, and there are like probably there are better targets, and we can find them during pathway analysis, and we will be able to find like more better targets in for gene therapy with a huger effect, and which will lead to like longer lives and maybe healthier lives, longer lifespan and health span. So when we're working with the gene expression data, almost always we working with differential expression between control and sample, or between like treated and untreated samples. And to do this, we have to have like two group of people, and we like we do many source. We can do many sorts of calculations. Our main goal to find like samples which are significantly different in their expression between groups, and to calculate magnitude of this expression. Sometimes not all of the users using it. And now most popular tools is DSEQ2, but actually it's not always the best option for like some users. And if you see an article that the guys used another one, or if they have done, they just we don't want to use DSEQ, better to like to understand why they didn't do this, because it could cause significant like performance loss if you use another technique, or if you will do quantile normalization. It's like quite often used, especially in CMAC project it was used, but it could like reduce. It could cut some signal from your data, and you have to be careful about it. And Actually, there are a bunch of data and a bunch of tools to analyze this data in RNA-seq, and it's overwhelming to understand which one of them better, which one of them worse. And quite a good review was published by Brienne et al., where they have shown that not methods performs equally good, and like actually all of these methods they have their limitations, pros and cons, and we have to assess and pay and keep in mind all their features. So overall, when we are talking about pathway analysis, there are like two types, but second type we can divide on like two subtypes. So first type is just analysis which preserves gene set, but don't preserve connections between them. And it's not always good, especially if we want to propagate signal through our network, or if you want to understand if it's effective. Another like methods, they call topology based methods, and they preserve all the connections, and sometimes even preserve the direction of connections and like how every genes affect its like downstream targets. Why is this important? Because sometimes we can see quite small changes, but when we propagate them through the pathway, and if it if it's placed in some entry point of the assay and affects all targets, it could be quite significant, even though overall amplitude will be not so huge. So what basically was done in the article, guys just have chosen 75 data sets for different diseases, and they have run multiple instruments in order to find pathways relevant to these data sets. For example, if it data sets from Alzheimer's disease, you just your tool will perform good if it will be like will have like relatively high rank, like first or second, and when it has like good p-value. And as guys showed, it's not always like possible through all pathways, analysis tools, and through all genes. The data sets overall ranks of target pathways was surprisingly high. If you are talking about median, so almost any tool was able to predict like 20s or like even 10 like median for like our data. And p-value also was quite high. So as you can see, the performance of these methods they are quite limited. But overall accuracy area under curve was not so bad, and especially for GCAA and for SPIA and Geronto tools. And if you are talking about topology-based and non-topology-based methods, topology-based performed a bit better. Then guys did quite important benchmarking, and it wasn't done almost like for any tool in the internet and for RNA-seq analysis. They checked its biases to null hypothesis testing. So basically, you can take all controls from all your data, from different data sets, and then randomly split them to control disease data sets, and you can generate a lot of these subsets and analyze them on different pathways and with different reasons. And what it does, it calculates how many genes has are prone to be like over optimistic or over pessimistic. I mean, if they are prone to like increase the number of significant pathway or decrease this number. And as you may see, the best tool in this case was GCAA. It had 0 BS pathway, and like also Geronto tools and SPIA was not so bad, especially in terms of in terms if to compare them with another tools, And for example, if you will take a closer look, some instruments which showed a good ranking for target pathways previously, they are not so good in terms of pathway biases, especially in biases toward zero, like for Purdue, which was quite good before. And you can see that this bias is uneven between pathways. Some pathways like tend to be more biased toward zero or toward one. One of the sources of this is the number of genes. So different pathways has different number of genes. And that's why if you have smaller number of genes, probably it's easier to be like predicted as significantly affected pathways, even though this is like pay attention to it and do normalization to a number of genes in the pathway. And most of the data were analyzed with the pathway with KEGG database. Actually, for GCA also can be used MC database, which has a lot of gene sets from different sources with different biological meaning. And when we are talking about pathway analysis, we have to keep in mind that it's not only KEGG, even though it's like implemented in almost all tools, but also wiki pathways and Rectome. And like this database contains an even bigger amount of pathways, but they have like different data structure, and you have to prepare them for analysis. And for some analysis, you can like prepare them with Hafite. It like works quite well, and it can process wiki pathways and Rectome. And with KEGG, it's kind of a bit problematically to download it from the website. You have to have registration and so on. And actually, last time when we applied for this for pathway licensing, we haven't obtained our letter, and we just used R, and R it's already implemented, and you can use it directly from R. And when you have different pathways, first thing which pop up is to combine them, and it already was done by Pathway Forte. And what guys did, they did it into like ways. One way was just to find similar gene sets, maybe gene sets which are fully overlapped, non-fully overlapped, and you can combine like super pathways. When everything is overlapped and everything is fine, probably it's most reliable pathways. Then you can like find some pathways with which have their analogs, and to do some sort of merging for them. And also you can preserve connections and directions between genes in your pathway, and it's like the best thing. But it's difficult to do this because it's not always feasible to understand how your weights and direction can be changed, especially when you're adding some things into your pathway. But overall, it was done, and it was generated like MPath database, and so now you're having additional database which are combining all three previous. It consists almost of 2,900 pathways, and like mainly from Rectome, but also like KEGG and wiki pathways there. And there are some subsets, and overall only 26 pathways were completely overlapped between three databases. And if you are using all these now it's four databases, like one generic database and plus, you can see that it's not always affect performance of the models drastically, but sometimes it affects. And what's interesting, it also depends on dataset. So you have like tool variability, dataset variability, and also pathway database variability. It's resources, and probably you would like to investigate all of them when you're looking with your data, because there are no like clear answer which one is better, especially when you're talking about databases. And what was interesting from the like observation of the article, there was like huge amount of inconsistency when some pathways, some pathway databases gives your result of upregulation, and another one for the same sample of downregulation of the pathway, or somewhere like significantly affected, and another one non-significantly affected. And also it was not only like for pathway analysis which preserves topology, but also for GCAA and Aura. We can observe inconsistency in results between different pathway databases. And as we remember, GCAA was most like precise thing. I mean, it had the least number of false positives, and probably if you even GCA showed inconsistent result, probably they are there, and like this pathway has different composition of genes, and it affects everything. So there will not be like easy answer for you when you perform analysis on C pathways databases. You have to be ready for this, that there will be like some inconsistency, and probably it will not be like maybe it's not even be possible or feasible to understand what is better, and we just have to keep all the answers and try to understand. Maybe we will have to filter out inconsistent results and save only like a coherent one, but it's not always will provide you a lot of data. You will filter out a lot of pathways. And speaking about GCA, one of the most it's not the most precise thing, it's like most conservative thing, I have to say. So it will not provide you a lot of pathways, but all pathways which it will provide you, they will be reliable. How it works, you calculate gene differential expression, and actually it's a tricky thing when you're working with GCA. It's well suitable for tasks like when you have patients and many groups of patients, like many patients in each group. For example, like 10 in control, 10 in pathology. But when you're working for something like drug treatments, when you have like only 3 replicates per one drug, it will be not so easy to do because GCA, when it performs own like adjustment and significance testing, it do permutation of labels of data, but it like it's a bit worse performed when you are using permutation of gene names, because you are disrupting intergene correlation. But still it works even with perturbation, and when you have like duplicates and that's it, you can extract meaningful information, but maybe you would like to use like additional constraints. For example, you can combine FastSA and Gauge, which like works with p-value more heavily, and it will provide you like smaller amount of pathways, but they will be more relevant. And a bit about how GCA works. You are performing differential expression. Some genes will be upregulated, some will be downregulated, some will be like unaffected. And you perform ranking from the like most affected genes from the most upregulated to like most downregulated. And for like GCA, you have to have a gene set. Basically, it's just some list of genes. You can download it from GCA, or you can generate it by yourself. Sometimes you have some set of genes and you want to analyze it. From which pathways it consists, and so on. So you have this gene set, and you are running it. You're calculating running sum. So if gene occurs in your gene set, running sum increased. If there are like gap, it will be decreased. And while this analysis is going, your enrichment score increases. While in most dense regions, and then it decreases when you don't see genes from your gene set. And basically, you normalizing then this score by division on the overall number of genes in gene set, and you can calculate. p-value and overall understand your pathway upregulated, downregulated, and if it's affected at all. And you have like huge number of subsets here, and in every disk category you can like, for example, for 2C5 you see there are like subcategories, and it's not an easy choice to like select one for you. So probably it's better to understand what are you investigated and which effects you're expecting from your perturbations genes or from in your like disease condition. It's like a typical output from GCA when you have like normalized enriched scores, and you have adjusted p-value for them. And adjusted p-value is also not so like obvious thing, because when we are calculating adjusted p-value for multiple hypothesis testing, we assume that our tests, they are independent, but they're not always independent, especially in pathways when you have like shared genes and when you have connected pathways, and it's like technique which used, but you have to keep in mind this stuff, that you actually your like tests they are not completely independent. And another tool which performed quite well, it was SPA, signaling pathway impact analysis. And why this tool matters? Because it preserves connections and directions between genes. And even sometimes when you have small like effects in your genes, it could lead to like drastic or at least significant perturbation of assay. What do I mean? For example, we have like the same pathway, and in one sample we have significantly affected B and E genes, and another we have B and A genes affected. And what we can see that F gene is like leaf gene. It doesn't have like any downstream genes, and so it's there are no signal propagation. And A gene, it's like entry point gene in the pathway, so it affects every gene. And even though for A gene we have like 1.5 fold change, and for F gene we have 4 change, yes, and overall perturbation is higher in like left case. Significance will be higher in right case, because we have like a bigger amount of accumulated changes in perturbation in our pathway, and that's why it's quite sensitive thing to work, especially when you're working with something which affects strongly your like phenotype, maybe some transcriptor factors or something like this, which has like low expression or which are hard to like dysregulate significantly. That's why SPA matters, and better to try to implement it in your researches. And it has also different metrics for significance detection. Overall, it's combination between a number of disrupted genes dysregulated, yes, and overall perturbation accumulation. And you can see that areas here like 1, 2, 3, and 5, they mean that you have statistically significantly affected pathways. The numbers here, it's pathways, and blue and red lines, it's different like thresholds after multiple hypothesis testing correction. It's FDR blue one, and red is one-for-one correction. But overall, we have to keep in mind this like overall dependency sync between some pathways in our data sets and pathway databases. Another good tool, which kind of it's more about unsupervised learning, is VGCNA, Better Genomic Correlation Network Analysis. and it's widely used, and it's a good reason for it. When you are using stuff like this, you can try to find some models which will not be biased by previous pathways, and as we have seen before, pathways can have like inconsistent information. So VGCNA quite good thing in these terms. And in VGCNA we have like many samples and gene expression for many samples ordered in a huge matches, and we are looking for correlation between genes. So we have multiple observations for every gene, and we are looking for correlation between them. Then we can like check. Also, we can check correlation between these genes and also try it if it is there, all between log p. Overall, we are using absolute values of correlation, so we don't take into account plus or minus correlation. Maybe it's not so good thing because we are using directions of our network, but still we preserve like all connections, and it's kind of a good thing. So and about correlation coefficients in the article, authors wrote that they implemented Spearman and Kendall-Tao, but overall, in the forums they wrote that actually with like from some time and experience they have seen that it doesn't work quite well, and they like just recommend to use Pearson or to use just before correlation from the original article. Actually, and also you have to. They don't recommend to use differential expression data. Yes, this is the case when we are using like on just normalized data, and variance normalization, which are the performance, is also like it could affect your pathway performance. So better just to start from like a default one, variance normalization. So when we like have our genes, we perform like all this variance normalization. Then if we have calculated correlation, we are building adjacent semantics, which are like just a correlation in some power, which we can extract from our data. I mean data in the analysis. And also, what significant thing to keep in mind, we have to be sure that our topology is scale-free, and we have to build it. So basically, when we have adjusted semantics, we are performing clustering, and from clusters combining k-means and hierarchical clustering, we can extract models. And then those models is like actually the essence of the analysis, like maybe some hanging fruit, low-hanging fruit, which can be then analyzed with some GCAE or like another tool. So you can send it to Cytoscape and see which pathways were affected, and genes in this pathway, maybe some like clusters of genes. And also important thing about VGCNA, you can find most connected genes in every clusters. They are on the fingers of MDS analysis. You can find them, and the genes which are most connected with in your pathway. So probably if you want to affect some pathway, you can start from these genes. And yeah, actually that's it. And as you may see, sometimes with the first running of this cytoscape analysis or something else, you can see that not such a huge number of genes are affected in those ways. So maybe your models they have like some biological meaning, and maybe also like starting some pathways. So sometimes better to like save them and to compare different experiments and different like samples, different experiments and their models between themselves. And it can like provide you good information on you can save this model like gene set, and you can then use it further like for GCAE or some other tool. So it's like some model relevant to your data, and probably you just want to save. And if to go in first in evolution of the methods, obviously we want to pay attention to GSAE. It's an autoencoder which has embedded gene sets. So basic input layer is your genes or like CPM or differential gene expression, and you have like second layer. You have gene sets. So as you may see, it's not fully connected. It has like just relevant genes connected to it, and it's called gene sets. And then you can combine these gene sets in superset, like in latent layer. Maybe I don't know. You would like to use like some additional layers, or maybe just some residual connections. It doesn't matter. Now we are talking about superset latent layers, and overall, the superset latent layers they keep information quite well, and you can compress your data quite good in this way. So if you calculate compression from these 200 supersets, you can see that overall information loss is not so huge. Maybe no, it's kind of big for Dunn index, but for other indexes, it's not so huge. And if you look on Disney graphs, they look like reasonable, and different cancer types they have their own color, and we can see that overall form and distances they are quite close. So probably it's a good method to like for dimensionality reduction, because when you have like 20,000 genes, it's quite a tricky thing to work with them and to understand what is the. And another good approach is like neural networks. You can compress your data into latent space, and then you can work with latent space and gene sets and try extract some data from there. Basically, you can do this also with pathways. And there are like approaches when you like incorporate your pathways in your latent space, and you can better for your like task. Better to start from different like autoencoders, at least from like the noisy autoencoders, they like shows quite good results for pathways. Like you will find more pathways in your latent space with them, and also with variational autoencoders. But if you will combine different like models and different approaches, you can combine their multiple latent spaces, and these multiple latent spaces they also contain a lot of information, and actually it's they can gather even more information if because some other methods they has their pros and cons. Autoencoders is kind of looks like a next step in pathway analysis, and we have to move to them. But it's not always easy to interpret these latent spaces, and we have to be careful about like some sort of overfitting and so on. And actually, they requires quite a lot of data if to compare with previous like examples. But still, we have this data. We have a lot of RNA-seq data in databases. The only thing they are generated with different instruments, different batches, and probably you would like to add this information to your like latent spaces, maybe to condition it by this data. There are like there are articles which like implemented some such approaches, and they show that overall results quite kind of good. And also we have to understand that for some like features it will be enough just simpler like one latent space and one method, and for another features we would like to use a full like battery of methods and latent spaces, and probably it's a bit more like more complicated states or features, and we would like to like extract all minor information and minor like factors, but we have to be careful with like additional noise and overall signals to noise ratio. Okay, so that's it. Thank you for your attention. I hope it was more or less clear.
Thank you, Dmytro. It's actually time for questions. Thank you for your speech. It's been great. So let's go for our questions. The first one we have here is First one is what is criteria to select pathway database? Sorry, what is criteria to select pathway database reference, or shall we include all available?
Yeah, it's a tricky thing, and there are no simple answer. Probably you would like to start from only one, but when you like perform some sort of analysis, you have to move first, or maybe you not have to, but you would like to, just in order to see. But you have to be like brave and be ready that there will be a lot of inconsistency and there will be no simple answers. That's why, like, my answer will be to use like all of them, but to be ready for pain with this, for mental pain.
To be ready for pain is a good answer. The another question is, we have all heard about the recent high-profile first-round investment of $3 billion in Altus Labs. What is your opinion? Does the technology have a future, and when we can expect rejuvenation technology and profound reversal of diseases like Alzheimer's for the masses?
Actually, can you repeat, because I missed the start of the question?
Sure. There was the recent high-profile first-round investment of $3 billion
Yeah.
in Altus Labs. So what's your opinion, like if the technology is ready?
Yeah, actually, they're working with epigenetic like reprogramming, and it seems like a good thing. From my experience, there are a lot of inconsistency too in epigenetic reprogramming, and we like kind of did some researches, and this biological age, it's not always so close, especially for mice, but for human too, which calculated by epigenetic reprogramming. But were a lot of articles which like inspiring articles, and probably it will work. And for me, it's like it looks like one of the like best directions of anti-aging medicine. When it will be ready is hard to say. It's definitely not ready. We don't have good approaches to reprogramming, especially like full organizing. We can reprogram cells. It's very quite like easy procedure, and but to like rejuvenate full organism, it's not so easy. Even though like there were like some experiments and kind of successful.
Oh, but do you think that this kind of investment can pay off in some, I don't know, 5 to 10 years period?
It's a difficult question because we don't know how this investment spread. If they have like few like good experiments and they are like working with them, probably yes. If like they spread it to like big amount experiments, it's maybe no. Because actually, I guess when we are talking about epigenetic programs, there are also a lot of white spots, and it's not always easy to do it like precisely and to tune it easily. And there are like they have to overcome a lot of obstacles. I guess I hope they will do it fast, but it's hard to believe that it will be fast.